afternoon, everybody. My name is Zhen, and my advisors are Dr. Hongqiu and Dr. Peng Liang. And the title of my dissertation is Kinetic Modeling and Analysis of FDG PET Imaging to Differentiate Malignancies from Inflammation and Brown adipose tissue. And my presentation can be divided into the following parts: statement of the problem, methods and results, summary, and future works. So first of all, I would like to introduce what is the problem we're studying and why it's important, which is the background, current limitations, and research significance, and our hypothesis and research framework. So let's first have a brief view of FDG PET. So positron emission tomography is a kind of molecular imaging technique which enables evaluation of biochemical changes within a living subject. And during PET imaging, an agent which contains a radioisotope is administered to the subject. It's called a tracer. And the positrons collide with the electrons to produce gamma rays with energy and travel at opposite direction. And it's called annihilation. And following that is the detection of the gamma rays by PET detector. And the annihilation events and the resulting electrical signals will be converted into sinograms and reconstructed into tomography images. And these images can reflect the distribution of the tracer inside the body. And for clinical diagnosis, PET is mainly used for detecting and staining tumors with the use of fluorine-18 fluorodeoxyglucose, which is FDG. And FDG has a half-life of 109 minutes, and it's an analog of glucose whereby the 2-carbon hydroxyl group has been substituted by a radioactive fluorine atom. So here are their differences. And the extent of FDG accumulation within the body is thought to reflect the glucose transporter activity. So FDG is taken into cells by GLUT1 and phosphorylated to form FDG6-phosphate by hexokinase. But it's different from glucose that further metabolism is prevented due to the absence of the carbon hydroxyl. So FDG remains trapped within the cell, and phosphate accumulates in the cells, leading to signal amplification, and therefore it's a suitable indicator for a cell's need for glucose. And the metabolic demands for the tumor cells are very different from normal cells. So FDG in tumor cells are significantly higher than normal ones, so it can be served as a biomarker for malignancies. And besides the FDG can have a high accumulation in the tumors, it can also have a lot of physiological uptake. And this is an example of our own data. And besides the subcutaneous tumor, we can also find high uptake inside the brain, the myocardium, the kidney, and the bladder, since FDG is not a tumor-specific tracer. And besides those physiological uptake, it can also be found in a lot of benign lesions. And there are two kinds of benign lesions that can cause huge false positive results, which are inflammation and brown fat. And inflammation is a kind of protective response to eliminate the initial cause of injury. And the activated inflammatory cells can cause the expression of the glucose transporter activity, and that is exactly the cause of false positive results. And here are two examples. The first one is the FDG uptake in different tissues in various time intervals. So we can see that inflammatory tissues uptake remain at a very high level. And the second one is the PET image of a tumor-bearing mouse with inflammatory lesions. The red arrow points to the malignancies and white one points to the inflammation. We can see from the image alone, we cannot differentiate the two kinds of lesions. And besides inflammation, brown fat is also a cause of false positive results. And it's a specialized tissue whose function is to produce heat, and it's found in mammals in cervical, interscapular, and periaortic regions. And it can help animals to survive the winter season without living on the shivering process to keep warm. And the proteins inside the mitochondria of the brown fat cells can consume the glucose for energy. So that's the cause of false positive results. So since FDG used as a non-tumor specific tracer can cause such serious false positive results, how do we restrain those results? And how do we differentiate malignancies from them? And are there any drawbacks of those differentiation methods? So here I have listed some current methods and their limitations, and hope to point out our research significance. there are some current methods to prevent the false positive results, and for inflammation, alternative tracers such as FET, radioligands, and FLT are not only tumor-specific, but also have a biomarker for cell proliferation. But FET is not easy to manufacture, and it's only available in rodents instead of humans. And radioligands, it has huge limitations due to the half-life of carbon-11 because it only has 20 minutes. And for FLT, it's also hard to manufacture, and not to mention its low image quality. And for brown fat, we can use food and temperature control or pre-medication to prevent this kind of effects. But serving food can cause physiological uptake, and temperature control, it's not realistic to avoid winter season for CAT scan, and pre-medication may cause side effects. So, at the meantime, a lot of researchers have proposed a lot of data acquisition and analyzing methods. The conventional acquisition method creates static PET imaging. It contains only one time frame scanned after tracer injection, and it's analyzed by a semi-quantitative parameter called standardized uptake value. This kind of method is hard to differentiate malignancies from those two lesions. And therefore, the dual time point PET image is introduced. It includes two static scans, and its principle is because the tumor's uptake increases with time while the inflammation's uptake decreases. But not all this kind of situation is like this, and there's not a gold standard for how much time that we should pause between those two time points. So that dynamic PET image and kinetic modeling was introduced. It contains multiple time frames, and the tracers are injected after the scan to make sure that the data is acquired and it can be counted as a function of time. And the parameters can be quantified by kinetic modeling, which I'll introduce in details in our methods. So here's our hypothesis. Our hypothesis is that kinetic parameters for dynamic PET images have a higher sensitivity to differentiate malignancies from inflammation and brown fat. And our research framework is listed here. At first, we established animal tumor and inflammation models and activate the activity of brown fat cells. And then we take two routes to verify our hypothesis. The first one is by dynamic PET data acquisition, and then we further analyze the data to calculate static and kinetic parameters. And at the same time, we do some histological tests for confirmation of that we successfully build the models. And at last, we take both imaging results and histological results for further correlation and verifications. So here's our novelty. The first one is that previous research didn't put emphasis on the importance of body locations. So we need to observe the influence of body locations of certain parameters. And the second one is that previous research didn't focus on the tracer kinetics of brown fat. So that we kind of observed the tracer kinetics of brown adipose tissue. So after introducing what is the problem and why it's important, I'm going to introduce our methods and results in detail. So, according to the research framework, our first step is establishment of animal models. Animal models are divided into tumor, inflammation, and activation of brown fat. And here's our experimental design. The tumor models, we picked the Lewis lung carcinoma cells and divided the animals into heterotopic and orthotopic group. And for heterotopic group, the tumors are inoculated subcutaneously, and for orthotopic group, the tumors are inoculated directly inside the lung. And for inflammation models, subcutaneous ones are induced the inflammation by turpentine oil on the right leg, and the in situ models are induced by dripping the LPS into the trachea. And for spontaneous models, the animals were put along with the mice infected with NHV to induce the liver inflammation. And for activation of brown fat, we did all the PET scans in winter seasons, and the animals were put for fasting for 18 hours before the scan to activate the brown fat activity. So here are our results. These two figures have illustrated how the tumors have grown after 2 weeks of inoculation. In the first one, visually we can see that the diameters of the tumor has grown to approximately 6.88 millimeters. And from the growth curve, we can see that after 7 days, the tumor starts to proliferate very rapidly, and after 2 weeks, the volume of the tumor has grown to about 320 cubic millimeters. And after the PET scan, we have extracted the tumors from the body, And both subcutaneous tumor and in situ tumors are very evident inside the body. And we also found the brown fat around the intrascapular region with a butterfly shape. So next step, we're going to perform the dynamic cut data acquisition. The dynamic data acquisition is approximately 16 minutes, and we extracted the last time frame, which is 45 to 60 minutes, for visual analysis. Visual analysis first. And the animals were divided into 6 groups, and here we can see that we extracted some of the tumor-bearing mice for inflammation inoculation, because we would like to observe the situation where both tumor and inflammation exists. So the groups are divided into inflammation with or without tumor. So here are our results. In the visual analysis, we can see that besides those physiological uptake, both subcutaneous and in situ tumors are evident. And the yellow arrow points to the subcutaneous tumor in the axillary area, and the red one points to the in situ tumor inside the lung. And for inflammation groups, the left arrow is subcutaneous inflammations. We can see that subcutaneous inflammation without tumor, it has a very high level of FDG uptake on the right leg, and it's much higher than the background, which is the left leg. And we found an interesting phenomenon that if tumor exists with inflammation, the uptake of inflammation is a little bit lower than the situation where inflammation exists alone. And for in situ inflammation, we can see that high level of uptake can be found inside the lung. And for spontaneous inflammation, we can see that an extraordinarily high level of uptake can be observed inside the liver, and it's much higher than the normal situation. And among all the groups, we have found that 4 mice have very high level of uptake in the intrascapular region. So we combine both septal and coronal images to find that this kind of uptake is caused by brown fat. So we did some comparisons, and we take in situ inflammation and tumor, for example. We can find that both tumor and inflammation has high level of FDG uptake inside the lung, and so did the brown fat. So we came to a conclusion that by visual analysis, we failed to differentiate malignancies from inflammation and brown fat only through the image itself. So we need to further analyze the data. And in this section, we have used both static and kinetic analyzing method for the data. And we also take body locations into consideration and separate the animals into 2 kind of groups. The first one is different lesions in the same body location, and the second one is the same kind of lesion in different body locations. And for static analysis, we extracted the ROIs and calculated the semi-quantitative parameter called standardized uptake value, and it's calculated in this form: radioactivity concentration in ROI over injected dose divided by weight of animals. And for kinetic analysis, we have used a 3-compartment model for FDG. And since I have previously introduced that FDG is taken by cells through glucose transporter and phosphorylated through hexokinase to form FGG6-phosphate. So this model is a mathematical form to simulate the biochemical changes of FDG inside the body. And the kinetics of FDG can be expressed in this form. Here, K1 to K4 are rate constants, and CPT is the plasma activity. CFT and CDT are the non-metabolist and phosphorylated FDG. And here, CTT is the tissue time activity, and it can be expressed here. And from the experimental data, we can observe CPT and CTT, which is the plasma activity and tissue activity, so that we can use experimental data to fit the equation and to estimate those rate constants, and therefore to calculate the influx rate constant, which have a physiological meaning to represent the FDG uptake inside the tissue. So here are our results. And for subcutaneous group, we found that both static and kinetic results showed significant difference between tumor and inflammation. And we found that inflammation with tumor its SUV is lower than the inflammation without tumor, and so did the kinetic parameters. And for in situ groups, we found that the static parameters failed to differentiate tumor and inflammation, but kinetic parameters succeeded in doing that. And for the same kind of lesion at different body locations, we found that both static and kinetic parameters fail to differentiate subcutaneous and in situ tumors. In our dynamic path acquisition results, we found significant difference between the time activity data of subcutaneous and in situ tumor. So we can differentiate those two kinds of lesions using time activity curve. And for inflammation at different body locations, we found both static and kinetic results can differentiate those groups of inflammation in different places. And for brown fat, although from static and kinetic results we found difference between those groups, but since our sample size is not big enough, so we need further verification to confirm our data. So in this section, we came to a conclusion that kinetic parameters have a higher sensitivity than static ones, and especially for in situ lesions. And the comparison of influx rate constant Ki can be a metric to assess inflammations at different parts of the body, because we found significant difference at inflammations at different body locations. So since now we have our imaging data, we did some histological tests for further verifications, like frozen sections and some staining, and here are our histological results. And we can see from the image that we found increased cell density and in tumor lesions, and it's a kind of, it represents the malignancies. And for inflammation at different body locations, we found inflammatory cell infiltration within the muscle fibers, inside the pulmonary lesions, and of course inside the liver, which represents inflammation. So based on our histological results as well as our imaging data, we found that those data can correlate and verify with each other. So to summarize, we first established the animal tumor inflammation models and activated brown fat activity. And then we did data acquisition and analysis for imaging data. And at last we did the histological test to confirm the image data, and came to our conclusion that kinetic parameters have a higher sensitivity, especially for in situ lesions, and the rate constant can be a metric to assess inflammations at different body locations. And for our future works, there are 4 points that we can do in the future. The first one is that we should reduce the acquisition time, because 60 minutes is a little bit too long for clinical application. And next, we would like to expand our sample size, especially for brown fat. And thirdly, we would like to study the mechanisms of tumor inflammation and brown fat. And at last, we would like to analyze the data of patients instead of rodents, because we want to seek the opportunity to apply our data into clinical application. So here's our thesis-related publications. And at last, I would like to thank my advisors, my team members, and my family members, and of course, your attendance. And thank you very much. And if you have some questions, I'd love to answer it.
Very impressive presentation. And I think it's open for questions and comments. I can ask you those questions. Yeah, you mentioned the traditional method may have false positive.
Yeah.
Yeah, you give us a lot of examples.
The static.
Right. So in your research, I didn't find any discussion there to talk about the false
Conventional
positive.
methods?
Yeah, compared to.
Yeah, I did some visual analysis and
But,
calculated the SUV.
I'm looking for the comparison between yours method to others in the literature. You give us several literature here. We have certain problems.
I have mentioned that in my introduction here, other literatures, if they have the static image and they cannot differentiate malignancies from inflammations, but I didn't list their SUV, and their SUV values are very alike, and so did my data. So we came to a conclusion that static image is not able to differentiate those false positive results.
Yeah, that's good, but if you could, in the discussion part, to give us quantity comparison between yours to others, that will be much better.
Yeah.
That's my suggestion.
And especially for if I tumor inflammation.
Yeah, especially for your submission to a general paper, they usually need you have a more deeper discussion, even between the others.
From my data and
Data, not only the
visual analysis.
Yeah. Usually they require such kind of analysis. For the master degree thesis, it's quite good already. I didn't see anything wrong. So any other questions from me? Because it's quite different with our background.
It's a combination of image data analysis with the biological tests.
If Professor Yang or other come here, maybe ask you other questions.
Because part of my work was supervised by Dr. Pence, so he's more familiar with the biological experiments.